Hello, everyone.
Hello. Hello.
Hi, gang. How are you today? Just a quick question for you in the chat window. I see your comment, Elena. Good to see that. Okay. Anyone have trouble with their internet connection this morning? Okay. All right. We got a couple yeses in there, don't we? Okay. So, yeah, I teach basically a 4-hour course in the morning before I teach you guys, and man, that really fouled us up. Lost internet connection from, I don't know, about 8:40 or so till about 9:20, and boy, it really threw a monkey wrench into things. So we'll do the best we can here, keep rolling. Okay, gang, we're going to get back into chapter 5 and get as far as we can. We need to be sensitive to our schedule here, right? Today is November 9th. Okay, and of course you have exam 3 the day we come back from Thanksgiving. So 3 weeks from today is exam 3. Okay. Okay, thank you, Brianna. I see your comment. Let's continue. Okay, so you may recall from last time, we were talking about chemical reactions that are really just part of a cell's metabolism. And we talked a little bit about collision theory to try to explain to you the circumstances necessary for a meaningful chemical reaction to happen. By meaningful, I mean we can either successfully create a bond or destroy a bond. And we also mentioned that, at least theoretically, we could increase the rate of chemistry by adding heat to a cell or the system in general. But that often is counterproductive because too much heat actually damages cells. It destroys the shapes of molecules. It denatures those molecules. And so Mother Nature came up with the idea of an enzyme. So we're going to continue from the point of talking about enzymes and then talk about carbohydrate metabolism in cells. Chapter 5 is a huge chapter. If you started reading it, you can tell that there's just a tremendous amount of content. And so perhaps I'll start with this statement. Okay, I've been looking through chapter 5, trying to find things that I can definitely tell you I will not test you on. And so I'm willing to make this statement, okay? In chapter 5, you are not responsible for material on photosynthesis, okay? You are not responsible for material on photosynthesis. I will not ask you any photosynthesis questions. It doesn't mean photosynthesis isn't important, but we have to pick and choose our battles with the limited time we have, okay? All right, so let's talk a little bit about enzymes, okay? Enzymes, as we said, are Mother Nature's answer to the heat problem, right? Too much heat damages cells. Okay, so let's take a look and describe what an enzyme really is. Figure 5.4 is probably a good talking point here because we talk about the components of an enzyme. Right. So, beginning biology students since the beginning of civilization, pretty much know that an enzyme is a protein. But that's not quite the whole story, right? An enzyme to a biochemist, and if I'm being frank, the way the biochemists talk about enzymes is the more scientifically useful way of talking about an enzyme. The way biochemists talk about an enzyme is that an enzyme is really a hollow enzyme, and hollow enzyme is suggesting that it is made of multiple components, okay? So a complete enzyme, a complete physiologically active enzyme is called a hollow enzyme. Okay, you see that in figure 5.4. A hollow enzyme is made of two things. Okay. Those two parts are the protein portion, which gets most of the credit, right? Everybody knows an enzyme is made of protein, so it gets most of the credit. Well, from a biochemistry perspective, that protein portion is called an apoenzyme. Okay, the protein portion of the enzyme is going to be made by the machinery of a cell, translating a piece of messenger RNA to make this protein, and that RNA is itself expressed from a gene through transcription, right? So the protein portion comes about through the natural process of gene expression. Okay, the other part is known as a cofactor. Okay, this is the non-protein portion of the enzyme. The non-protein portion of the enzyme comes about through nutrition typically. In other words, you typically eat foods and drinks that contain material that can act as a cofactor, okay? There may be some circumstances where a cell will manufacture a cofactor through other chemical pathways of its metabolism, but we often ingest these things, okay? And there's a little bit of nuance here. I'm going to say it this way. So a hollow enzyme is made of an apoenzyme, which is the protein portion of the enzyme, and either a cofactor or a coenzyme, okay? A cofactor or a coenzyme. Your textbook in figure 5.4 uses the phrase cofactor, but we need to qualify that statement a little bit, okay? So the term cofactor refers to an inorganic component. And by inorganic, I mean we're talking about anything that does not have carbon, no carbon present, right? So examples of this would include metals like iron, zinc, things like this. When I say metals, I'm talking about the metals of the periodic table, okay? Elements that are categorized as metals, right? So if we have an inorganic component to the hollow enzyme, that inorganic component is referred to as a cofactor. Alternatively, you can also have some hollow enzymes that actually have something that's technically not a cofactor, but is instead a coenzyme. Okay. The term coenzyme refers to an organic component. In other words, carbon is present. an example of a coenzyme is something that we're going to encounter later in our conversation in this chapter, such as a molecule that by its short name is called NAD. And there's different versions, there's different oxidative states of NAD. But for right now, just call it NAD. NAD is a molecule that is short, the full name for NAD is nicotinamide adenine dinucleotide. Okay, it's a mouthful. Nicotinamine adenine dinucleotide. Let me make sure that I'm saying that properly for you. Okay, hold on just a second. All right. Let me Google that because I don't want to get that wrong for you. Okay. Yeah, nicotinamide adenine dinucleotide. That's what it is. Okay. So nicotinamide adenine dinucleotide is a fairly complicated structure that has carbon atoms in it, and therefore it would fit the definition of a coenzyme. Okay, so whether we're dealing with an iron atom as a cofactor or a molecule of NAD as a coenzyme, both of these things act as a copilot to the apoenzyme. They help the apoenzyme do its job better and fly the plane correctly. Okay, and so I don't know what your experience was growing up. Everybody has a different experience. But when I was a little boy, my parents, they wanted me to take some vitamins because I didn't always eat the most healthy foods. I didn't eat the best. And so every day I would take a Flintstones vitamin, right? A supplement that you could buy at the supermarket, and it happened to be in the shape of Fred Flintstone, right? So the point of it is, if you're taking, like Centrum or one-a-day vitamin supplements, then they are going to have the cofactors and sometimes coenzymes that the apoenzyme portion of our holoenzymes need to function correctly, and that helps support a healthy metabolism so our bodies work correctly and everything's happy here. The same is certainly universally true of the metabolism of bacterial cells, fungal cells, protozoal cells, you name it, the basic chemistry is the same. Okay. All right. Okay. Let's continue here. So now we know that a full enzyme is properly known as a holoenzyme. The holoenzyme is made of some fancy words called apoenzyme and cofactor or coenzyme. Okay. Let's talk about some of the properties of enzymes. Okay, and if you've had this in other courses, this should sound familiar to you. First off, we know that the main purpose of an enzyme is to accelerate a chemical reaction. To make a chemical reaction happen faster. There are some chemical reactions inside our cells that if we did not have an enzyme to give that reaction a kick in the behind to go quicker, that reaction would take the rest of our natural life to complete itself without the enzyme present. If that were the case, you and I would not be sitting here having a conversation because life would not be what it currently is now. Okay. So next point. Most enzymes are made of protein, right? So, but we know more properly the holoenzyme business up above. Exceptions exist, okay? And I'll just mention the one that we talked about, right? Ribosomal RNA is a piece of catalytic RNA, meaning that there are times when RNA can behave as an enzyme. At least in this chapter, we're really not going to deal with that all that much. Okay, next. Enzymes are not consumed by the reactions that they make happen. In other words, an enzyme can make a reaction happen, and then it can go off and do it again. So the enzyme can be recycled many, many times before it wears out. Okay, so enzymes are not consumed by the reactions they make happen. Enzymes can repeat their function millions of times before wearing out. Okay. With some exceptions that I won't talk about at the moment, with some exceptions, enzymes are generally speaking very specific for one single substrate. Okay, so in other words, an enzyme that's designed to break down a glucose molecule really is not going to break down a lactose molecule. It's not going to break down a maltose molecule. It's not going to break down an amino acid, right? It is specific for one kind of molecule. Enzymes are specific for one type of substrate. They do not generally modify other molecules. Okay. Enzymes can be recognized by their names, right? Which is to say the end of an enzyme has the phrase ASE. Enzyme names end with quote unquote ASE, right? If I could type properly, that would make sense. Okay. So the name of the enzyme that can break down lactose sugar in human cells is called lactase, right? ASE. The name of some enzymes that move phosphate atoms around are called kinases, KINASE. The name of other enzymes that add phosphate atoms to certain molecules are called phosphatases, ASE on the end. The point I'm trying to make is that enzymes are named with the ending ASE. There are a couple enzymes that were identified before the naming convention of applying ASE was adopted by the scientific community. And so there are a couple really historically old exceptions. And unfortunately, my old brain I'm not remembering one of those exceptions off the top of my head. But most enzymes today, 99.9%, can be recognized by the ASE ending. Okay. Next, enzymes do their job. They accelerate the chemical reaction by lowering what's known as the activation energy. Okay, activation energy is actually something that is hinted at in your textbook. Let me give you a figure on that. Figure 5.2. Enzymes work, which means to say they accelerate the reaction by lowering the activation energy of the reaction. Okay, that references figure 5.2. So we should probably at least define activation energy. Activation energy is the amount of energy you need to get the reaction started. That's what it is, okay? Perhaps another way of, well, I could define this multiple ways. Activation energy is the amount of energy that must be applied to make meaningful, and when I say meaningful chemistry, I mean create a bond or destroy a bond, make meaningful chemistry happen, okay? Another way of communicating activation energy is to say activation energy is the amount of energy needed to permanently disrupt the electrons between the atoms that are interacting to either create a bond or destroy a bond. Alternatively, the amount of energy needed to permanently disrupt the electrons around the atoms that are interacting. Okay. All right. So without wanting to get into too much nitty-gritty biochemical detail here, basically what enzymes do is that they grab onto molecules and stabilize their motion long enough for the correct atoms between the molecules to actually bump into each other, interact, and break or add bonds. Okay, so I hope you remember last time we talked about this, we were talking about the nature of molecular motion, that molecules translationally move, they rotationally move, and they vibrate. All of these motions increase the probability that molecules will collide, but not collide in a meaningful way, because they're just bumping around all over the place in this complete chaos. So what enzymes do is that they bind to molecules and they slow down the amount of vibration and rotation in them long enough so that when the molecules collide, they can collide with the proper orientation and relationship to each other so that the chemical reaction can actually succeed. Okay, so it's kind of like having, it's kind of like having 11-year-old boys and girls together on a playground, and they got crushes on each other, but they don't really ever have any meaningful interaction. And then, a playground attendant comes over and brings them two together and forces them to join hands a little bit, and then they smile and grin and do all that kind of good stuff, right? That's basically what an enzyme does. It kind of acts as an intermediary to make something meaningful happen. Okay. Enzymes stabilize the motion of molecules long enough so that bonds can be created or destroyed. Okay. All right, folks, any questions so far? All right, so that's just a little bit about enzymes, and hopefully that's not too horrific for you. Okay, so we've covered a lot of what your textbook is talking about. As we get into the meat of this chapter, we got to pick and choose our battles. Okay, so this chapter of your book talks a lot about carbohydrate catabolism, which is to say the breakdown of sugars to extract energy in a way that can be used to build ATP and so on and so forth. Your book also talks about photosynthesis. That's important, but I'm choosing right now to not make it a focus of our lecture time. Okay, so you do not have to study photosynthesis to do well on the upcoming exam. Okay, so we need to talk about enzymes and how they function in the context of carbohydrate metabolism. That's what we need to focus on for the remainder of our time together. And I suppose since I'm at the end of a page, it just makes sense to go down to the next page and start fresh here. Okay. Okay, so let me just ask ask one or two questions here to prime the pumps in your brain, okay? So before I do that, let me show you another document of which I'm going to share with you later today in my email that I'll send you. When we talk about carbohydrate metabolism, the conversation is wickedly complicated. And so I'm going to be sending you this little summary sheet here. Okay. Here's an overview of carbohydrate metabolism. that we could talk about. And honestly, if we talked about these in a proper biochemistry course, would probably need at least a full month's worth of lectures. And we just don't have that kind of time. But let me just read through this diagram with you and give you some perspective on what it is I'm going to ask you to understand. So we'll start here. Different cells under different environmental conditions use different chemical pathways to break down different carbohydrates. I mean, if you take a moment to think about it, that seems fairly reasonable. Different cells, whether it's a bacterial cell, a fungal cell, a human cell, plant cell, whatever it is, different cells under different environmental conditions, right? You might have a soil bacterium living in the soil outside your front yard, or you might have a bacterium like E. coli living in your large intestine right now. So their environmental circumstances are different. Different cells under different environmental conditions use different chemical pathways to break down carbohydrates. Generally speaking, there's three broad groups of reactions we can start with. First is something known as the pentose phosphate pathway. This would be used by a bacterial cell such as Bacillus subtilis. You're not really going to be tested on this. I wouldn't spend a lot of time on the pentose phosphate pathway. You've got enough to worry about. There's an alternative pathway known as the Entner-Dewotteroff pathway, which is again is an alternative way of breaking down sugars. This is performed by Pseudomonas aeruginosa, which is another kind of bacterium, right? Not really going to challenge you on that either. Okay, not that it's not important, but we only have so much time to deal with things. The version of carbohydrate metabolism you have probably been introduced to, in which I'm going to expect you to have a fundamental basic understanding of, starts with glycolysis, okay? Glycolysis is the most common way, at least the most common way from what we know as scientists, what we've studied and cataloged from life on the planet. It is the most common way for cells to catabolize carbs, which is to say to break down carbohydrates, okay? It's the most common way that we have discovered when we survey life on the planet. Most cells seem to do this, with some notable exceptions. So we start with glycolysis, which we will be going through later, probably even today a little bit. And we can break from glycolysis, we can enter two alternative pathways that take place in a cell after glycolysis finishes its work. That could be cellular respiration. And by cell respiration, that has a very specific meaning to a biochemist. That means that there is a Krebs cycle present and an electron transport chain. So if you see a Krebs cycle and an electron transport chain as part of the conversation, you immediately know we're dealing with cellular respiration. The other chemistry that can take place after glycolysis is fermentation, right? And many of us enjoy alcoholic beverages if we're 21, right? So a nice glass of wine or a beer from time to time. Fermentation takes place after glycolysis, just like cell respiration. But if fermentation is going to take place, it does so without a Krebs cycle or electron transport chain. In other words, fermentation as a catchphrase for chemistry that happens after glycolysis involves chemical reactions that do not use a Krebs cycle or electron transport chain. So let's finish out this part of the pathway here. Fermentation, no Krebs cycle, no electron transport chain. Now, okay, remember I had that one question on the one workbook assignment recently, and I was asking you, is oxygen present during this experiment? And many of you said no, because it's fermentation and it doesn't use oxygen, right? And then we said, oh, that's an assumption, so we got to be very careful with it. Well, here's the idea. Fermentation can definitely take place when oxygen is absent, okay? When oxygen is absent. And that scenario is going to be involved even in your own body. Your own cells will ferment when there's an oxygen debt. So think about the last time you winded yourself through exercise, okay? Hopefully that's because you just ran 5 miles, not because you just stepped off the sofa, right? So I'm teasing you, but I'm out of shape too. So fermentation can take place in the absence of oxygen, such as when you exhaust yourself through exercise. It's also used by microbes such as lactobacillus, which performs lactic acid fermentation. Fermentation also happens in the absence of oxygen by Clostridium difficile, which you know, famous for the C. diff infections in hospitals and so forth. And it's also done by baker's yeast, Saccharomyces cerevisiae, that makes alcohol. What maybe you are not as familiar with is that fermentation can take place in the presence of oxygen, but still be ignored. So I mentioned to you at the workbook activity that cancer cells will do this kind of fermentation. Oxygen's available, but the cancer cells, because they're abnormal, choose to perform fermentation. This form of fermentation also takes place in plant pollen and also takes place in some pathogens like Trypanosoma brucei. Trypanosoma brucei, you might know or recall, is a protozoal parasite that causes African sleeping sickness. Okay, so we can have glycolysis followed by fermentation. The fermentation may be in the absence of oxygen or the presence of oxygen, depending on the specific cell involved and the circumstances the cell is exposed to. An alternative to fermentation, and which is very much the more common form of energy production in our cells and in most bacteria that we would think about, is cell respiration. And as I said, that always involves a Krebs cycle, and it always involves an electron transport chain. What might be a shock to you is that it's not uncommon that I get beginning students that always tell me, oh, well, if there's no oxygen around, then that means it's fermentation. No, that doesn't mean that. It doesn't mean that at all, right? You can have cellular respiration that's anaerobic, okay? And by that, I mean there's no oxygen available. And by that, I mean something other than oxygen is the terminal electron acceptor in the electron transport chain. This would be the scenario used by species of archaea, which we haven't talked a lot about in our course, and some bacteria such as Disulfovibrio vulgaris. The most common way of thinking about carbohydrate catabolism is that it's typically aerobic, so oxygen is the terminal electron acceptor in the electron transport chain. This is used most times by normal cells of your body, okay? In times when there is not an oxygen debt, it's done by plant cells, and it's done by, I would say most species of microorganisms that we're aware of. So this is an overview of carbohydrate metabolism, and you can see that you could quickly get into the weeds and be like, oh my God, this is overwhelming. What do I need to know? Okay, so folks, here's what you need to know. For purposes of examination, what am I going to hold you responsible for? Folks, here's what I'm going to hold you responsible for, okay? Hopefully my graphics pen will work. You need to know glycolysis, cell respiration, which is aerobic. Okay, this is the piece of the carbohydrate metabolism puzzle that you're going to need to carve out of Chapter 5 and do your best to understand it. Okay, that's not an easy task because this is a lot of stuff. But this is what I want to hold you responsible for. This is what I want to try to teach you. This is what I want you to have some knowledge of, okay? Thoughts or questions so far? Okay. All right, folks. So, like all complicated journeys, you got to start somewhere, right? Like the journey of 1,000 miles begins with a single step. Okay, so by George, I don't even remember what question I was going to ask you. That's horrible. Oh, yes. Okay, here it is. What is the purpose of aerobic cellular respiration? What have you thought about, heard about, talked about in other courses? Yasmin has a question in the chat window. You will be sending that to with our notes? Yes, Yasmin, I will. Okay. So let's be clear here. You are responsible for understanding glycolysis, Krebs cycle, electron transport chain in the presence of oxygen. So aerobic conditions. Okay, so it is my hope that you've had at least a little bio background so you can tell me something about this question. Okay, so, all right, because our time is limited, I'm going to run with what you feed me. Okay, so Rachel says to form ATP. Rachel, I agree with you. Elena, you say to release a large amounts of energy in multiple steps. Elena, I agree with you. Send important things to maintain healthy cells and tissues. Samantha, I agree with you too. I would be curious what you mean by important things. And Chelsea, you say ATP production. So, okay, we'll go with what seems to be the general statement that I'm getting from your chat comments, and that is to manufacture ATP. Okay, to manufacture ATP by extracting energy out of food. And when we say food here, I'm specifically talking about glucose, right? Sugar, okay? So that is certainly a huge and a big, important and correct answer. So that is correct answer one, okay? There's something else that we typically fail to introduce undergraduates to simply because of the lack of time in our courses. But I want to mention it to you because I think it's something that you should appreciate, okay? And so I'm going to ask you in your textbook, please have your textbook open. Please take a look at figure 5.33. Figure 5.33. It's really at the back of chapter 5, okay? The back of chapter 5. Okay, and so let me just be sure that we're all on the same page. I want to ask you a question. What is the title for figure 5.33? What is the title of figure 5.33? Okay, thank you, Elena. It is the integration of metabolism. Let's think about that just for a minute. The integration of metabolism. What the heck does that mean? Well, what I'm suggesting to you and what your textbook is trying to communicate to you is that carbohydrate metabolism in terms of glycolysis, the electron transport chain, the Krebs cycle, all of that good stuff, which you can see referenced in figure 533, right? And if you go to the top of figure 5.33 in the center, doesn't it start with glycolysis? And as we go down, we see that green little arrow that's in a circular shape, and in the center is the Krebs cycle, okay? So the point I'm trying to make is that these processes that we associate with making energy are also used by cells to do other very important tasks, okay? And so without wanting to spend too much time on it, but to at least mention this to you, what I'm trying to suggest to you is, well, let me ask this in the chat window. What are the 4 categories of organic macromolecules? What are the names of those 4 categories? What are they? Come on, folks. Dig deep. Scrape out the cobwebs. Okay, let me help you because I want to move forward, right? Carbohydrates is one. Okay, good job, Samantha. Carbohydrates, lipids, proteins, and nucleic acids. That's what I'm getting at. Good job, folks. I know you can do it. All right, so if you look carefully at figure 5.33 in the upper left corner, do you notice that there's a purple rectangular box with the word nucleotides in it? Nucleotides. Additionally, on figure 5.33, if you're on the right side of the image and you carefully scan your eyes gently below, eventually you get to an orange colored rectangle with the phrase fatty acid in. Huh, that's curious. Continue moving your eyes down further and you find a blue rectangle with the phrase amino acids in. Okay. And so what I'm trying to tell you is that carbohydrate metabolism, which is famous for producing ATP, also drives the chemistry of manufacturing nucleotides to build DNA and RNA. The chemistry of metabolism of carbohydrates actually drives the synthesis of fatty acids needed to make lipids. The chemistry of carbohydrate metabolism also drives the synthesis of amino acids used to make protein. And if you look very carefully, like if you look carefully at the phrase amino acid in the bottom right corner of figure 5.33, do you notice that to the left of the phrase amino acids, you have a double-headed arrow? So in other words, there are things that leave the Krebs cycle to build amino acids. And in some cases, amino acids can go to the left and inject themselves into the Krebs cycle to help the Krebs cycle do its job. So the take-home message I'm trying to give you right now is that the chemical reactions of carbohydrate metabolism, which are famous for making ATP, also enable the cell to manufacture the building blocks for all four categories of organic macromolecules. Okay, let me say that again. The chemical reactions associated with carbohydrate metabolism support the synthesis of building blocks, nucleotides, monosaccharides, fatty acids, amino acids, the building blocks of all four categories of organic macromolecules. Those building blocks, if they are found in a cell, because the cell just absorbs something out of the bloodstream that you ate, those building blocks can be injected into the chemistry of carbohydrate metabolism to get energy out, or in some cases, the chemistry of carbohydrate metabolism can leave the traditional pathways of Krebs cycle and so forth to actually synthesize nucleotides, fatty acids, amino acids, monosaccharides, okay? So there, figure 533 is the integration of metabolism. Carbohydrate metabolism is very similar, to use a very rough analogy, this would be like Grand Central Station in New York City, right? Maybe some of you have been in the rail network in New York City. Grand Central Station, right? This hub of rail activity that all the other trains come into and out of Grand Central Station, right? Carbohydrate metabolism, glycolysis, Krebs cycle, electron transport is Grand Central Station. for all of the building blocks of the cell. Okay. So, we're not going to take time to literally describe this chemistry because that's way beyond what our course can do. But, folks, please give me some feedback. Am I making sense here? Do you see what I'm talking about? Would you like to ask any questions? So all these complicated chemistry, which I'll be honest with you, I know that most traditional students don't like this topic because it is chemical in nature. And so it scares you if you haven't had a chemistry course. We're not going to do fancy things like balanced chemical equations and stuff like that. We're not ready for that in this class. But what we can do is have a conversation about these topics and make sure we get the big picture of why these things actually matter. Well, carbohydrate metabolism matters because we can build ATP. ATP matters because it's kind of like an energy storage battery that cells use to power things like moving vesicles around inside the cell, along the cytoskeleton, or powering muscle contraction, or creating a nerve impulse in your nervous system, right? That requires ATP. So we need this battery that we call ATP. How do we get the battery? We manufacture it by extracting energy out of glucose and other molecules that come down the pipeline, okay? So energy production is huge, but also have an appreciation that the energy production chemistry of figure 533 supports the manufacturing of additional building blocks that are important for the cell to live and be what it actually is. Okay. All right, gang. So enough talking here. We're going to have to dive off the deep end and start talking about these processes. And so I'm going to show you a video at this time on glycolysis, Krebs cycle, electron transport chain. And then I'm going to start walking you through these pathways in chapter 5, and I'm going to start asking you questions to see what you know and what you don't know. Okay. YouTube video. Okay. On aerobic cellular respiration. Let me get the link, put it in your notes.
How do you
get this played in just a moment? Okay. All right, so now you have this video that you can study on your own during your personal study time. Let's watch this, see what it has to say to us, and then we'll go through some of these pathways together. Okay, and I think this is a good video. Unfortunately, it's not quite high definition, so it might be a little bit blurry at times, but that's the price you pay for getting free content, right? Free material. So here we go. Let's have a look at this and see what we can do here. And okay, here we go.
How do you turn that bite of food into a chemical that a cell can recognize and use as energy? The first step is altering the food into its component chemical compounds and then getting those molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that bite of food into useful energy by cellular respiration begins. The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream. From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a 4-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of two distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, two ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the two 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistants. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain. The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane. When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain. All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that entered the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use. This process starts with the eating of a snack or meal and ends with capturing the energy from the complete breakdown of the nutrients into energy and carbon dioxide. How do you turn that
Okay, folks. So there you've seen it, the mystery of life. We gave you a summary in 6 minutes, but it is a pretty respectable video. And we're going to use that as a foundation to walk through these pathways together, okay? Are there any questions you'd like to ask just after having seen the video one time? Okay. All right. All right. So again, you have the link there in your notes. You can review again. And of course, there's many other cell respiration videos freely available on YouTube if you want to do a little searching through that, okay? So what I'd like to do next is literally start talking to you about these pathways, but rather than me sitting here and describing this happens, then this happens, then this happens, which is terribly boring and not very useful for you, what I want to do is a question and answer session where we look at these things together. I ask you questions. If you get them right, fantastic. If you don't, then I explain the answer to you, okay? So let's start question and answer session on glycolysis. Okay, so this is figure 512. Figure 5.12. Okay. That's horrible. There we go. Okay, folks, let's see what we can do here to pull back the fog that might be in front of your eyes, okay? So I'm going to be asking, everybody's going to get a turn at a question here, okay? So don't be scared, don't be embarrassed if you answer incorrectly. This is a friendly place and we are here to learn, okay? So, okay. All right. So let's see what we can do here. Okay. Aubriana, can you please say hi to us? Aubriana, are you okay? Can you unmute your microphone and say hi to us, please?
Hi.
Hi, Aubriana. How are you? Are you okay?
Yeah, I'm good. How are you?
I'm okay. Hey, do you have figure 5.12 open in front of you? Can you see it?
Yeah.
Okay. All right. So Aubriana, I'm going to ask you a question here. Okay. So Aubriana, go up to the top of the page where you see figure 5.12. And obviously I'm asking everybody to consider this, but right now only Aubriana is going to attempt an answer. So you see there's different chemical reactions that are color coded with an orange ball, like orange ball 1, orange ball 2, orange ball 3, the whole way down to the bottom. So Aubriana, at orange ball number 1, okay, can you tell me what the name of the substrate or substrates is in that chemical reaction?
Is substrate that's the enzyme, right?
That's what I'm trying to get you to figure out.
It's just the product is glucose 6-phosphate. What does that mean?
So I'm just typing out the question in the notes and giving you a little time to process. Okay, so what are the names of the substrate or substrates at chemical reaction number 1? So pretend we're on a game show on television, and I'm going to ask you, Aubriana, what's your final answer here? What are the names of the substrates at chemical reaction 1?
I don't know. Is it ATP?
Okay.
ATP?
ATP is one correct answer. Is it the only correct answer?
And ADP?
No.
ATP and ADP?
No,
And
not
glucose?
both. No. Okay. All right. All right, Aubriana. Okay, well, this is game show. So, okay. So.
Oh, it's pyruvate. Is it pyruvate?
Pyruvate is not the substrate at step 1, but
Okay.
Aubriana, let me give you a way to redeem yourself. Can you tell me the definition of a substrate?
No, not off the top of my head.
Okay. All right. Thanks, Aubriana. Obviously, that's something you do want to figure out before your exam, right? So, okay. So let's help Aubriana out here. The correct answer to my question is glucose and ATP. Glucose and ATP are both substrates. Okay. So let's make sure we define what a substrate is. Okay. A substrate is any substance that is modified by the enzyme. Any substance that is modified by an enzyme. Any substance, and any molecule that is modified by an enzyme. Okay. So folks, you can see here, I didn't ask Aubriana to balance a chemical equation or anything like that, but I am asking that everybody in the class be able to answer questions like these that I'm about to pose to you, okay? So the substrates at chemical reaction 1 are glucose and ATP. A substrate is any molecule that is modified by an enzyme. Okay, all right. Question 2, okay. All right, next person up to bat is Afala Shadi. Afala Shadi, please say hi to us.
Hi.
Good afternoon, hope you're doing okay.
Fine.
Afala Shadi, here's your question. Afala Shadi, at chemical reaction 1, what are the name or names of the products of chemical reaction 1?
The products is the ATP and the ADP.
You are partly correct. Do you want to change your answer?
And the product should be the ADP.
Okay, so you are half correct, Afala Shadi. ADP, D as in David, ADP is an answer. Is there anything else that's a product?
I just noticed the ATP is constant everywhere.
Okay, all right.
Okay,
Shadi,
do you mean the glucose phosphate? I think that's going to 2.
Okay, I can work with that. Thanks, Afala Shadi. I appreciate your participation today, right? So a product is what is going to be manufactured because of the activity of an enzyme, right? So the products in this case are going to be ADP and glucose 6-phosphate, glucose 6-phosphate. Okay, so we want you to be able to tell the difference between a substrate and a product if you look at a chemical pathway. Okay, notice we're going in a very precise order, right? We're starting at number 1 and we're progressing down. We're progressing down the page. Notice the direction of the vertical golden yellow arrows, right? That's implying there is a directionality to this chemistry. Okay, next question. All right, next question. Okay, so Alexis Pearson. Alexis, could you say hi to us?
Hello.
Hi, Alexis. Alexis, are you able to see figure 512?
Yes.
Okay, great. All right, so here's my question to you, Alexis. Alexis, what is the name of the substrate at chemical reaction 2?
The glucose 6-phosphate.
Okay, so stay with me, Alexis. You're doing fine. So Alexis, you're saying it's glucose 6-phosphate, and I agree with you, but there's something here I'm trying to get across to everybody. So Alexis, wasn't glucose 6-phosphate the product of the previous reaction?
Yes.
Okay, so if that's true, Alexis, what principle am I trying to illustrate with my questions?
In order for chemical reaction 2, it needed the product of chemical reaction 1.
Beautifully done, Alexis. Thanks for participating today. Okay.
Thank you.
Okay. So gang, what I'm trying to show you here is that the product of chemical reaction 1 becomes the substrate for chemical reaction 2. And so the general idea here is that the product of a previous reaction becomes the substrate for the next subsequent reaction. That is how biochemical pathways work. You build one thing, that thing that you built is the product, and then that product is fed in as a substrate in the next chemical reaction that happens after the first reaction. Okay. Notice that in biochemical pathways, that the product of one reaction becomes the substrate for the next reaction. Okay. All right. All right, folks, you're doing okay. Let's keep going here. Okay, let's see, that was question 3. This is question 4 coming up here. All right, so next, Chelsea Benzinger. Chelsea, please say hi to us.
Hi.
Okay, Chelsea. So Chelsea, can you see figure 512?
Yes. Yep.
Okay. And so Chelsea, on figure 512, you notice that there are 10 orange balls indicating 10 chemical reactions.
Yeah.
So Chelsea, here's my question to you. What is present at each orange ball? What is present at each orange ball, that is not literally printed in your textbook?
Would it be oxygen?
Well, let me tell you something. Glycolysis does not require oxygen.
Oh my goodness. Okay. Is it? It's not a sugar. An
So,
enzyme.
I'm sorry, say again?
Is it an enzyme?
Yes, it is, Chelsea. Good job. Okay. All right. So I guess what was that? 3 attempts? That's not too bad. Okay. So thanks for participating. You did fine. You know, here's the question. What is present at each orange ball? In other words, each chemical reaction, but not actually printed in the textbook. And that is an enzyme. Okay, here's what I'm trying to get you to see. Okay, you know, textbook publishers try to reduce the cost of printing their materials by sometimes abbreviating things and making assumptions about what they think students already know when they look at this object in the textbook. But the point of it is here, each chemical reaction of glycolysis, each chemical reaction of basically all of the carbohydrate metabolism we're going to talk about depends on the presence of an enzyme, a specific enzyme for a specific chemical reaction. Okay, so what is present at each orange ball is an enzyme that enables that chemistry to take place, even though that enzyme is not stated in figure 512. Okay. All the chemical reactions of aerobic cellular respiration, rely on specific enzymes to carry out specific tasks, okay? And so, let's see here. I want to do something with you. Okay, I want to do something with you. So let's have a little race here, folks. I want to have a little race online with you. I'm gonna type a challenge to you in the chat window, and the first person to get the correct answer is going to win the race. What do you win for the race? Well, you win my hearty good job and a clap, okay? Here's what I'm asking for you, okay? Here we go. I want you to locate the full glycolysis pathway at the back of your textbook. What is the page number in the upper left corner of that page? Go ahead, folks, find it for me. Find it. Put in the page number in the upper left corner of the page that you identify. It's at the back of your book. I'll even go so far as to say it's in one of the appendices. Okay, Yasmin, it looks like Yasmin has probably won it. I don't know if there's a difference between the online digital version of the textbook and the physical book that I have. I can tell you that the physical book that I have, the first page of the full glycolysis pathway is on AP-2, AP-2. Okay, but if you found it in the back, yeah, you found the right thing. And the point is that your textbook has the full pathway printed up, including with the names of enzymes, right? So let's do this in the chat window. Here we go. Here we go. What is the name of the enzyme that makes chemical reaction number 8 happen? What is the name of the enzyme that makes chemical reaction number 8 happen? Okay, just use those pages you just found and locate chemical reaction number 8. Tell me the name of the enzyme. Okay, there it's coming up. Good for you folks, right? It is phosphoglyceromutase, phosphoglyceromutase. Are you going to have to memorize that for your exam? No, you do not. But if I gave you information and asked you what's the name of the enzyme, I'd expect you to be able to pick the enzyme out, not memorize it, pick it out, okay? All right. Folks, we're just getting started with questions here, but as things would be, we are out of time. So expect that next class, I will do my best to finish chapter 5, and most of finishing it is going to be asking you questions to see what you understand and what you don't understand, because there's no way you're going to get this if we don't walk through it, okay? So do you have any questions right now? Okay, folks, have a nice afternoon. It's warm outside. Go take a nice walk. I'll see you on Thursday.
See ya.
Thank you. Bye.